Good morning, afternoon, evening, or middle of the night, depending on which your time zone is. My name is Professor Paul Collinson. I'm delighted to chair this 2024 IFCC live webinar on troponin assays with higher clinical specificity, a novel future option. We have a distinguished faculty for you today with Professor Allan Jaffe, Sarah Whittooth, and Stephen Meeks, who are going to talk in order about this rather fascinating and novel concept. I'm going to kick off with Professor Allan Jaffe, who is the Wayne and Catherine Priesel, have I got that right, Allan? Professor of Cardiovascular Disease Research. I've known Allan for over 40 years, which is a terrifying thought, and even more worrying, he's known me. I would first, though, like to congratulate Allan, who's just been awarded the 2024 IFCC Cardiac Biomarkers Prize for his distinguished contribution to cardiovascular biomarkers. He's currently chair of the Division of Clinical Laboratory Services at Lab Medicine at the Mayo, and also a consultant in the Division of Ischemic Heart Disease. So he's one of those rare breeds. He's both a lab person and a cardiologist. So on that note, I'm going to pass you over to Allan. Allan, please begin.
Thank you, Paul, and thank you for your kind words. If I might have my slides on, please. Hopefully they are on, because I can't see them. Nonetheless, we have a tendency to think of troponin as simply a peptide of, depending upon the particular analyte that we're talking about, the peptide may be 23.5 kilodaltons for troponin I or 33.5 kilodaltons for troponin T. And we think about that as if it's just one protein that comes out into the circulation. Next slide, please. One looks at this cartoon that was developed some years ago by Alan Wu. It depicts a variety of fragments, however, and this is just a small percentage of them. And I want to spend a couple of minutes because we need to understand how these might be developed. So one thing that is thought to be the case, and may or may not be 100% true, is that early on after the development of an ischemic injury, free chains come out first, whether it is troponin I or T, and that it is later that big hunks, TIC complexes, IC complexes, are released. Now, some of this is potentially not as well studied as it would be ideal, because many of these early studies, because they used Western blotting techniques, required fairly high values. So we're talking about bigger infarctions, many of the patients treated with lytic therapy or reperfusion therapy. And after reperfusion therapy, it's clear that TIC and IC complexes are more apt to occur. In addition, it does appear that one can have free circulating troponin I and C and form IC complexes. And it's also possible that TIC can degrade into troponin T and maybe a CT complex. So there are a variety of different protein fragments that potentially could be measured. Next slide, please. And if one looks, and this is a typical example from the literature utilizing, again, older data with less sensitive assays, so you can ignore the specific troponin values, showing that one has an upper band of troponin I, which is a little bit higher and may represent one of those larger fragments, intact troponin I, and then degradation fractions of a variety of sorts. And we'll talk a little bit about what those are in just a couple of minutes. But you can see that over time, the intact protein is degraded, and it may well be, therefore, that there is some information either in the degradation products, as I'll show you in a couple of minutes, or by being able to identify early on the intact, initially released protein. Next slide. Now, this is just again, a slide from the literature that looks early after the onset of a given event and suggests that most of what is seen there is intact troponin I in this example. By the way, I'll show you one example of troponin T, but I'm not going to focus nearly as much on troponin T, not because it doesn't do something very similar, but because Sarah Whitford is really the world's expert on this and has been working in this field for some time. And I'm sure she will show you some interesting data about that. Next slide. And this is later. And you can see that over time, degradation products then are developed as troponin is degraded. And troponin is cleared almost exclusively by degradation and by fragmentation, not necessarily, as some people have averred, through renal clearance. There is a very tiny component, if any, that is cleared renally. Next slide. So, are there really free chains first? Well, maybe. Hold on. Let's go slowly. It's not totally clear, and it may well be that those free chains, some of those free chains come from degradation of the TIC complex, or if IC is released, and there probably is some IC released, although it's not as much as TIC, that could come as well as those are defragmented. In addition, this may change very much with reperfusion. And one needs to be aware, therefore, that it's not totally clear once one reperfuses, in particularly with a lytic agent, which has more protease activity, that these fragments became much more complex. And then you have all the degradation, and the question is which of those fragments are detected. Most of the assays, as I'll show you in a couple of minutes, that are used commercially are developed by trying to detect as much troponin as possible. So it tends to target all of the common fragments. On the other hand, if one has degradation of some fragments where the epitopes for one or the other 2 or 3 antibodies is removed, one could miss those degradation fragments next. And as this is, again, large events, because we're using Western blots to detect them, and that requires a lot more protein. And one of the issues that we'll have to talk about eventually is some of these fragments are a relatively small percentage of the overall troponin that is released, and therefore one is going to need more and more sensitive techniques in order to be able to probe any of these. This is an example of troponin T. Again, higher molecular weight, 29.5 kilodaltons is probably the primary one. What you can see is eventually there is degradation shown below to smaller fragments. Troponin T, in one sense, has an advantage in the sense that the commercial antibodies are very close to each other in terms of the number of amino acids they are apart. So even the smaller fragments are detected more readily, whereas with cardiac troponin I, the epitopes are further away, so one could miss more fragments next. This is another important issue to keep in mind. These are data from Jennifer Van Eyck, done years ago, looking in ventricular tissue per se. It would be ideal if what happened is that nothing happened in the tissue and everything happened in the bloodstream, and then we could look at each one of these fragments as if it is unique and processed only in the bloodstream. However, that's not the case. Troponin I is processed in the tissue if it sits there for a period of time. This comes from a surgical example, but if one had a totally occluded coronary artery where there is relatively little perfusion, we think there is a little bit of perfusion, but not very much in a totally occluded artery, the troponin is likely processed within the ventricular tissue. So the timing of when one looks for these fragments becomes critical. Next. And this is just a slide to remind me to show you that most of the assays, and this is a very old slide with older assays, but the principle is the same. Most of the assays work hard to detect all of the fragments so that they give you the biggest troponin signal possible rather than looking at specific fragments. Next. So, one possibility. Next, go forward. If one could detect the forms that are released early, say free chains, might one have a more sensitive probe for the diagnosis of myocardial injury? And we'll talk about types of myocardial injury in a minute. The caveat to that is timing. If there has been an event or part of an event, and we know that sometimes events can be stuttering, some of this could represent processing within the myocardium per se, so it may not be pure. In addition, release of all these forms is blood flow dependent. So if one has an open or partially open coronary artery, as most patients with non-STEMI have, one may see a different panoply of fragments being released than if one has a totally occluded coronary artery and needs to reperfuse. Nonetheless, it is attractive to think that if we could have an early marker to detect one of these pure forms, that maybe we would have an earlier marker of myocardial injury. Next. Now, it gets a little more complicated and gives us an additional opportunity as well, because some of these fragments can be described, and this is again from Jennifer Van Eyck, who showed initially that the first fragmentation that occurs, at least in the myocardium, is at the carboxy terminal end, such that one ends up with a carboxy terminal depleted form of cardiac troponin I, 1 to 193 shown below. That seems to be the first cleavage. What's interesting about that cleavage, next slide, is that it may be unique to a specific pathophysiology. And in this particular instance, I'll show you in just a moment, if one overexpresses that 1 to 193 peptide in experimental animals, one ends up with a cardiomyopathy with calcium handling properties that is reminiscent of stunned myocardium. Again, timing will be key. Next slide. So one could have direct myocardial injury that might give you different forms. Next. This is an example of that, and hit one more because this is animated. you can see that if you give dobutamine to healthy volunteers with and without coronary disease patients as well, and again, I hit forward one more because it is animated, or it should be. There it is. Even in normals, there's a tiny little bit of release in troponin. This is due to direct myocardial injury caused by dobutamine. Does that release a different set or a different portfolio of forms than acute ischemic heart disease? There are no data to answer that. Next slide. Next. Okay, myocarditis could do something very similar. Inflammatory responses of the myocardium are very likely to change the processing of all of these fragments, so that it is conceivable that we could end up, assuming timing is on our side, being able to develop a portfolio of fragments that would be more specific for myocarditis. There also is a preload-related myocardial injury, next slide, that has been documented by Joe Canty. That is to say, when one has acute increases in preload, volume expansion being one, probably this is the same mechanism when one has exercise. As one increases pulmonary pressures, one has acute increases in preload, first in impedance, and then because of RV dilation in the RV, and one releases troponin. The troponin is proteolyzed and released. Next slide. And the cells die due to apoptosis. Again, this may be a mechanism where having very different sets of fragments might give one a mechanism or more in-depth information. Again, the percentage of the fragments that we need to be looking at is going to be critical, and we're going to need very highly sensitive probes to do that. Next slide. So with these, we might be able to detect different possibilities. I put in Takotsubo's cardiomyopathy because Takotsubo's cardiomyopathy is one of stunned myocardium. And Jenny and I have speculated in print that if we could interrogate the carboxy terminal peptides and fragments that are released, that might be a hint to the diagnosis of Takotsubo's. But as many of you know, the troponin signal in Takotsubo's is usually not large. The percentage of carboxy terminal protein is relatively small. So that, again, there's a very substantial issue in regard to sensitivity. Next slide. Well, yeah, and this just goes back to Jenny's slide to show you the 1 to 193 and the next. And this is the cardiomyopathy that is induced when one transfects this abnormal protein into experimental animals. And if you do that, you end up with something that, at least from the point of view of calcium handling, looks like stunned myocardium, and perhaps therefore might be a hint to Takotsubo's. Next. So overall, whether or not this is increased sensitivity by seeing free chains, or whether or not we can be more precise in regard to specific disease entities by looking at fragment analysis notwithstanding, this gives us an opportunity to markedly expand the information that troponin gives us. The caveats we need to be careful about are that timing is critical, as I showed you, in particularly given that things can be processed within the myocardium. We need to be careful about the mechanism, and unfortunately, clinically, often the mechanisms are not pure and only of one etiology. And the third issue is we need very, very highly sensitive probes, far better than the blotting techniques that we're using. But there is the ability to develop better antibody as well as a variety of other techniques in single molecular counting to improve sensitivity. So eventually we probably can get there. The question will be, how do we mine this, and how do we then use this information to help us clinically? Thank you very much.
Well, thank you, Allan. That was really food for thought for us from the clinical perspective. I'm going to move on from Allan's talk now, which is essentially looking at what the clinical problem is, to Stephen Meeks, who is at the Cardiovascular Research Center in Maastricht, where he's a clinical chemist and a cardiovascular scientist at Maastricht University Medical Center with a primary interest around cardiac troponin. And he's going to talk to us now about navigating towards a more specific cardiac troponin assay. Stephen, over to you.
Thank you very well, Paul, for this introduction. Yes, the title of today's talk for me is Navigating Towards a More Specific Cardiac Troponin Assay. I will start with my disclosures. I received research support, honorary and consultancy fees from Roche Diagnostics and Abbott Laboratories, all paid to my employer and reinvested in research. So the large majority of currently used troponin assays are so-called high-sensitive assays, and that name speaks for itself. Even very low concentrations of cardiac troponin circulating in the blood can be detected, and clinically that sensitivity translates to a very high negative predictive value of almost 99.5%. That is a great feature for safely ruling out people who present with chest pain. A possible disadvantage, however, is that those high sensitive assays can not only detect troponin in patients with acute myocardial infarction, but also in the majority of healthy individuals or a wide range of other conditions, cardiac and non-cardiac. And that limits the specificity of the test. And it's the reason why the positive predictive value is often suboptimal, somewhere between 70 and 80%. A patient group that is particularly affected by limited specificity and positive predictive value is the elderly patients and subject with chronic kidney disease. So my story today is about the exploration of a strategy that may improve the specificity of the troponin assay without compromising the excellent sensitivity. This illustration nicely depicts the continuum of cardiac troponin concentrations measured with high sensitive assays from healthy individuals towards myocardial infarction. And two key features of troponin measurement determine specificity: the concentration at presentation, with higher troponin levels increasing the probability of myocardial infarction, and the presence of a substantial rise and fall during serial measurements. An interesting fact about circulating cardiac troponin TNI, also mentioned by Allan, and which is not captured in this slide, is that cardiac troponin circulates in the blood in various forms and sizes. Circulating cardiac troponin is in fact a mix of intact troponin and troponin fragments of various sizes. The first one that is prevalent is TIC complex. The second one is intact troponin T. The third one is troponin T where the N-terminal is chunked off, and the fourth one is troponin T where both the N-terminal and the C-terminus are missing. Well, this is actually a simplified view of what is really happening. In practice, it is basically a continuum of all possible fragments of possible sizes, but for comprehensive purposes, I now limit it to these 4 main forms. We hypothesize that detailed understanding of the fragmentation pattern of cardiac troponin T could provide an opportunity to increase the specificity of the assay without affecting the sensitivity of it. I now will take you to a number of experiments that were conducted to identify the fragmentation of cardiac troponin T. This is a typical release curve that is observed when you measure troponin T in a patient that presents at the cardiac emergency unit. And here on the y-axis, you see the troponin concentration, and on the x-axis, you see the time after presentation. What you see is typical rise and fall up to 72 hours after the patient presents with chest pain. Now, these are numbers obtained with the clinical assay. If you would measure troponin with Western blot, which would allow you to measure, to identify the size and the fragments of the cardiac troponin, you will get a graph like this, a typical Western blot. Now, I know that not all of you will be familiar with this kind of experiment, so I will run you through it. Here is a positive control, the marker, and the negative control. And here is time 0, and then all the way up to time 72 hours. What is apparent in this Western blot is that the early hours after presentation are characterized by large intact cardiac troponin T, primary fragments, and secondary fragments. So these are smaller than the intact troponin, and the secondary fragments have a size between 14 and 18 kilodalton. What is also apparent is that early after presentation, you see a relative abundance of intact troponin and primary fragments, and in the aftermath of the myocardial infarction, the secondary fragments predominate. That also shows you that if you measure cardiac troponin at 2 time points, you basically will measure a different set of molecules. Another technique to basically demonstrate differences in sizes over time is column chromatography. Also, this is something that not everybody may be familiar with, so I will guide you through it. Here on the y-axis again, the concentration of cardiac troponin T, and on the x-axis, you see the volume of eluted fractions from the column. This technique works in such a way that the large fragments elute early from the column and the small fractions elute later on from the column. And when you measure cardiac troponin levels in all these fractions, it is apparent that the 3 hours after myocardial infarction, the sizes are predominantly large and a relatively smaller number of small fragments. When you expose the same 13 patients to this experiment, but then with blood not drawn 3 hours after myocardial infarction, but 15 hours after myocardial infarction, you see a shift in the size of cardiac troponin T. After 15 hours, the large fragments is substantially reduced, and the smaller fragments are the dominant fragments in the mix that you measure. How does this compare to other settings where troponin elevations are common? And a very interesting one is chronic kidney disease or end-stage renal disease. A group when they present at the cardiac emergency can often lead to clinical confusion. When column chromatography is conducted in these patients, you see that actually the fraction that is only visible in this patient group is the fraction that corresponds to the secondary fragments, being the very small troponin T fragments. There's no intact troponin, there's also no primary fragments. And that is the case both in pre-hemodialysis and post-hemodialysis. A similar pattern is visible in another setting where troponin is elevated, and that is exercise-induced troponin release. When you conduct column chromatography, only secondary fragments are visible in these subjects. So what these data suggest is in myocardial infarction, you have a mixture of intact troponin, primary fragments, and secondary fragments. And there are groups of patients where there are only secondary fragments apparent. Although this cannot be discriminated with the classical clinical assay, because they target all the fragments, both intact and all the way up to the very small fragments. So to understand how this fragmentation exactly works, a number of experiments were conducted. Here you see schematically the troponin T molecule and the clinical assay that is used in the various laboratories with the antibodies directed against the middle portion of the troponin T molecule. If you would measure 2 patients with myocardial infarction, here indicated with sample 1 and sample 2 with Western blot, and in this Western blot, the clinical antibody is used to stain the blood. What you see then is, in this case, no intact cardiac troponin T, but you do see the primary fragment and you do see the secondary fragments. The positive control, which is spiked cardiac troponin T in troponin negative plasma, gives a signal at the intact troponin and a small signal at the primary fragment. Now, if you do exactly the same experiment, but you don't use the clinical assay, but you use an antibody directed against the N-terminal of cardiac troponin T, is what you will see is that sample 1 and sample 2, the 2 patients with myocardial infarction that here gave a nice signal, are basically empty. So that means that this part of the molecule is chopped off early after myocardial infarction. The positive control, as you can see here, is positive as expected, and the negative control is negative. So this experiment led us to conclude that the N-terminus is one of the early events in the fragmentation of troponin T. Then Sander Streng in our lab spent a number of years using mass spectrometry to very carefully assess what the exact composition is of the various fragments. And in line with the previous experiments, he showed using mass spectrometry that the 37 kilodalton lane here in the Western blot comprises, as you would expect, from all the parts of the troponin T molecule can be recovered in the mass spectrum. The primary fragment, in line with the previous result, lacks the N-terminal part of the troponin T molecule. And the secondary fragment lacks both the N-terminal part of the troponin T molecule and the C-terminal part of the troponin T molecule. And that brings us to the following interesting summary. When we divide the fragmentation in roughly 4 categories, so the first one being TIC complex, the second intact troponin T, the third one troponin T where the N-terminus is lacking, and the fourth one troponin T where both the N and the C-terminus are lacking, then we can conclude that in myocardial infarction, all these forms are present and can be detected. In other settings, like for example end-stage renal disease, post-exercise, but also in some other settings of chronic troponin release that are not discussed today, we see that those groups are limited to only the secondary fragments. Now, the scenario right now is that the current clinical assay detects all these forms. It is conceivable if you redesign the assay in such a way that this form would no longer be detected, is that you create an assay that remains sensitivity for myocardial infarction, but gets rid of the troponin signal in chronic kidney disease and after exercise. And the next speaker will go into this in much greater detail, but what it comes down to is we now know that the fragmentation occurs first by cleavage of the N-terminus and then by cleavage of the C-terminus. But if we would change the assay in such a way that this antibody is replaced from the mid portion to the C-terminus, that you hypothetically would get an assay that no longer detects the secondary fragments while preserving the sensitivity for the troponin that is released during myocardial infarction. And that's where I would like to end and return the word to Paul.
Well, thank you very much for that, Stephen. That gives us some major food for thought, and I hope we can have some discussion about that in a short while when we have the attendee questions. But I think you've given an excellent lead-in now for Sarah Whitruth, who's the Assistant Professor of Biotechnology at the Department of Life Technologies at the University of Turku. And University of Turku actually has a tremendous track record in developing the understanding of how troponin assays work. And every paper that I've ever seen that's come out of there has always been fabulous and well worth reading over the years. So on that, I'll pass you over to Sarah. I think you're on mute, Sarah.
Oh, my apologies. So now I hope you can hear me. Yes, and I will start sharing again.
Fine.
Yes. So good. So I can get started. So thank you very much, Paul, for the kind introduction, and I will continue with my topic. So my topic is novel assays for measuring different molecular forms of troponins. So I will continue where Stephen has led us, and I will give you more insight into the immunoassays. So as we have heard already from Allan and Stephen, troponin is present in the circulation. It can be present in many different forms at the different stages of degradation. But most of the studies have traditionally been made with Western plotting techniques or gel filtration, or in some cases also with mass spectrometry. These have provided us very important information on the degradation of troponins. But if we're considering that the aim is to develop better methods for diagnosis or for clinical care, we need simpler methods. So the Western blotting and gel filtration methods, they are very complicated, and as Allan already mentioned, their sensitivity is limited. We need something more sensitive and something simpler. So let's consider immunoassays. Of course, in immunoassays, we are talking about antibodies binding to target molecules. So first of all, we have there the antibody, capture antibody that is attached to a solid surface. Then that antibody binds to the target molecule, and then there is the other antibody that is labeled, the tracer antibody, and the label produces a signal that can be measured. And this all can be automated quite easily, and actually is the basis of all the current clinical laboratory troponin tests available there in the clinical laboratories and market. And also high sensitivity is achievable, as we can see with all the high sensitivity troponin tests there available at the moment. So if we consider immunoassays for cardiac troponin T, I have here the simplistic picture of the structure of the protein. We have the N-terminal end and C-terminal end shown. And as Stephen already showed, there are places where cardiac troponin T is cleaved. There are the major cleavage sites. First of all, there's the N-terminal cleavage site between the amino acids 68 and 69. And then there is the actual cleavage area towards to the C-terminal end. spans from amino acids 189 to 223, and there are actually multiple cleavage sites located in that area. So the current assays that are being used at clinical laboratories for measuring cardiac troponin T, for example, the Roche cardiac troponin T assays, the high sensitivity troponin T assays, they are designed so that the antibodies are binding the stable central part of the molecule, as Stephen actually already mentioned, and this means that they are actually detecting, well, first of all, the intact full-length troponin T molecule, the primary fragment that has been cut at the N-terminal cleavage site, and then also the secondary fragments of a bit different sizes depending on which cleavage site they were cut at the C-terminal cleavage area. So, as Stephen already mentioned, in renal patients and in marathon runners, it has been detected that only the small fragments, secondary fragments, are found in their circulation, while in myocardial infarction, the predominant form at the early stages are the long forms, either the intact or primary fragments. So we want to target these myocardial infarction specific forms, and as Stephen showed, that could be done by using antibodies that bind to, well, one should bind to the stable central part, but the other one should bind to the C-terminal part. And as both antibodies have to be bound for the molecule to be detected, the result is that with an assay that is designed this way, we can only detect the intact form, full-length form, and the primary fragment, but the small secondary fragments are not detected because the molecule is cut between the binding sites of these antibodies. So we actually developed such an assay in my laboratory, and we published the first results in Circulation in 2022. And we actually used this kind of approach in our work. So high sensitivity is very important for the method, as highlighted already by Allan. So actually, instead of using one labeled antibody, one tracer antibody, we decided to use 3. So in this assay, we have one capture antibody that binds to the C-terminal part of the molecule, but then we use 3 different labeled antibodies. It is enough of one of these antibodies to bind to produce signal, but all the labeled antibodies can actually bind at the same time to the same molecule. And this way we get more signal and higher sensitivity for the assay. I will just go a couple of slides back because I just realized that I forgot to mention an important point here. So for the Roche assay, as it's or similar troponin T assays, as they are targeting the central stable part of the molecule, they are kind of detecting all the major forms of troponin T found in the circulation, which means that we actually can also call this assay a total troponin T assay. So it kind of measures all the important forms there. So it measures the total troponin T. And now I get back to where I was. So the results are what we get, or we have published with this assay. Well, first of all, we studied patients who had end-stage renal disease, so severe renal dysfunction, and also we investigated patients who had non-ST elevation myocardial infarction. And the samples were taken from these patients within 24 hours from the symptom onset. And if you look at the figure here on the left side, you can see the results from the total troponin T assay. So this total troponin T assay here is the Roche high sensitivity troponin T assay, which measures all the major forms of troponin T in the circulation. We can see that in the renal patients, in the ESRD patients, the concentrations are clearly elevated, and they are actually at the same range of concentrations what we see also in the non-ST elevation myocardial infarction patients. But when we analyze the same samples with our assay for long cardiac troponin T forms, we saw that with infarction patients, their concentrations were still elevated, but for renal patients, the concentrations were very low. So we actually confirmed the previous results that it seems that in renal patients, the circulating troponin consists of the small fragments because there is very little long forms of troponin present in their samples. What we also did is that we calculated a so-called troponin ratio. So this ratio is calculated by dividing the result that we got with the long cardiac troponin T assay with the result that we got from the Roche assay. So we'll divide the long troponin value with the total troponin value. And with this, we get a number that kind of reflects the fraction of the long cardiac troponin T forms in the sample. So for example, on the left side, you can see the representation of the non-ST elevation myocardial infarction patients. So for those patients, this ratio was 0.29. So this means that roughly 29% of the circulating troponin in these patients is actually in long form. So 29% of the total troponin T is in long form. While on the right side, you can see the situation in the renal patients, in the ESRD patients. And in these patients, the ratio is very low. In our study, the median ratio was only 4%. So only 4% of the circulating troponin T is in long form. So we get back to the same figure that we looked at already on the left side, but now I have added here on the right side also the ratio results. So if we compare on the right side the ratio values in the renal patients, in the ESRD patients, and in the infarction patients, we can see that actually this ratio separates these groups very well, even better than just measuring the long forms. So we can present the ratio results in another form like this. So using this ROC or receiver operating characteristics curve, and the solid line here is the troponin ratio. So we can see that in this form, the area under the curve for the troponin ratio, or the fraction of the long forms, is 0.955, while the area under the curve for the Roche test for total cardiac troponin T is only 0.609. So the test can, or the ratio can much better discriminate between these two groups. We also looked at the troponin levels in time after symptom onset. So here on the left side, you can see the total troponin T concentrations in infarction patients at different time points. Each dot represents different patient and samples taken at different time points after the symptom onset. And as you can see, quite naturally, in the early hours, the concentrations are quite low because it takes time for the troponin to end up in the circulation. But after that, the concentrations seem quite variable. While on the right side, you can see the same results with same patients, but for long troponin T. And here you can see that at the early hours, the concentrations are, of course, again, quite low. But then there seems to be a peak of concentrations, and then the concentrations come down. When we use these data to calculate the troponin ratio, we get this kind of results. So actually, there's very clear trend of the ratio being high at the early hours, and then it starts to decline. So what this figure tells us is that even though at the early hours the concentrations of total troponin and long troponin are quite low, still of that low value of total troponin, actually very high fraction is in long form. And when the time goes on, the ratio becomes smaller. Always smaller fractions are of the long form of troponin. So this also means that this test would be most useful at the early hours. So let's say within 24 hours after the onset of symptoms. This also means that this kind of test could be useful in diagnosing reinfarction, because the concentration or the level comes down relatively rapidly. But within 24 hours, it should work. So yes, we got quite promising results with this assay that we developed with these first samples that we studied. But these were actually quite extreme patient groups. There were infarction patients who had quite high concentrations. The samples were taken actually quite late after the onset of symptoms for many patients. And also we were dealing with renal patients with very severe disease. So actually, when considering the clinical use of such assay, it would be most interesting to find out whether or how well does this assay work in early samples, and especially in non-ST elevation myocardial infarction patients. So that is an important question to be studied in the future. And also, in addition to renal patients, it would be good to know how this assay works in the other patient groups who typically have elevated cardiac troponin T, or the special occasions of which Allan mentioned in his talk. But to study these groups, well, if we consider the early samples of non-ST elevation myocardial infarction patients, the concentrations can be quite low. So we definitely need high sensitivity. So if we consider the high sensitivity assays that are used in a clinical laboratories right now, for example, the Roche high sensitivity assay that measures all the major forms of troponin T in the circulation. With the long assay, we are measuring only subpopulation of those molecules. So actually, even better sensitivity is required for a long cardiac troponin T assay. The assay that we used in the study that I showed to you, well, it had better sensitivity than the traditional methods like gel filtration or Western blots, but still the sensitivity is not that good. As you can see here, the limit of detection is 11 nanograms per liter and limit of quantitation 25 nanograms per liter. So we have worked with this assay, and actually we have been able to develop even more sensitive form of it. So by utilizing a special label technology, which is upconverting luminescence, which means that actually we use these particle labels that are able to combine the energy of 2 photons into one high energy photon that is emitted and measured. So that allows us to produce very highly sensitive assay with the limit of detection and quantitation levels that you can see on the slide. So limit of the quantitation is 1.8 nanograms per liter. So now we are at the level where we can actually study also those patient groups and individuals who have only low elevations of troponin. One important to consider when using when using the long cardiac troponin T assay for studies is that you should not use serum samples. Why is that? Well, thrombin enzyme is activated in serum when serum samples are prepared, when the blood is clotting, and thrombin also degrades troponin T, especially at the N-terminal part, but also at the C-terminal cleavage area. So one should not use serum samples. So yes, we have got promising results with the approach that was also presented with Stevens to use antibody combination that detects the troponin forms that are not degraded at the C-terminal cleavage area. But are there other possibilities to target other molecular forms of cardiac troponin T for better sensitivity? Well, one that is comes to mind is the use of the N-terminal cleavage point and screen for molecules that are not degraded there. I have not seen any published research on this, but as implied in Stevens' presentation, it seems that the N-terminal part is very unstable and cleave very rapidly. So probably it would not be that feasible to use this approach. How about then troponin I? There are major cleavage sites also in troponin I. What makes situation difficult with troponin I is that actually the current immunoassays available for troponin I use very different approaches with the parts of the molecule that the antibodies target. Here are some examples that are used in current high sensitivity troponin I assays. This information is from the biomarkers reference tables that can be found at the website of our committee, the IFCC Committee on Clinical Applications of Cardiac Biomarkers, or the CCB Committee of IFCC. But as you can see here, there are many different options. With troponin T, it's easy as we can compare to the reference. All the assays have the antibodies targeting the middle, certain part in the middle part of the molecule. But for troponin I, it can be more complicated and difficult to show the benefit. And also the furthest parts towards the N-terminus and C-terminus, they are the most unstable. So it probably would not be that useful to target those. But there is no published data of similar studies as have been now conducted with troponin T. So we can wait and see if in future there will come some up. But I would like to summarize my part here, just reminding you that the methods that were previously used for analyzing the troponin T fragments are complicated and they have limited sensitivity. But immunoassays can allow better sensitivity and can be applied to automated systems. There's some promising preliminary data on the immunoassay for long cardiac troponin T forms because it can discriminate between the infarction and renal patients, and the ratio of long cardiac troponin T and total cardiac troponin T seems to work even better. And now there is high sensitivity assay available. This is a research assay. It's not commercial and it's not automated, but there's a research assay available. And then for the other possible approaches, well, at least there doesn't seem to be any published data yet. So by this, I conclude my part. So back to you, Paul.
Thank you, Sarah. That was absolutely fascinating. I really enjoy a lecture where I'm really being having my preconceptions challenged, and that was one that was absolutely perfect in that regard. The we've got a few questions have come in, and I've got a few of my own actually, but I'm going to kick off with one question which I think is entirely relevant. Which has come in, which says, why do we use the term high sensitivity troponin? And I think it's quite important to clarify here, when we talk about high sensitivity troponin assays, we are just literally talking about the ability of the assay to detect above signal above noise ratio here, and that we're not detecting a different form of troponin, we're just detecting it more sensitively. And the IFCC committee has recommended the definition of high sensitivity assays, one which you can detect with a CV of less than 10% at the 99th percentile, which in real terms is actually the female 99th percentile. In fact, is often much lower than that anyway, and where you can detect at least 50% of a healthy reference population. So I will deal with that. And the question I'm going to actually put to Sarah and Stephen at this point is, troponin T and troponin I are essentially similar but slightly different. And one of the things that we've always known about troponin T is that in chronic myocardial injury, it tends to give a greater signal than troponin I, and that it is a better predictor of long-term total mortality, whereas cardiac troponin I tends to be a better predictor of cardiac mortality. Now, looking at your final slide, Sarah, I can see that this might be something to do with which assay is looked at, but do you both think that this reflects the fact that troponin T actually assay, as it's currently configured, picks up more troponin?
Well, maybe I start then, but yeah, well, it's hard to speculate, but I could say, I could think so, because it's detecting more of the different forms that could have something to do with it. Do you have a comment on this, Stephen?
I'm not sure whether it picks up more troponin. What is interesting, however, in respect to prognosis is that my expectation would be that the prognostic aspect of cardiac troponin T is particularly captured by the smaller fragments, because that is the part that is elevated in chronically elevated patients like chronic kidney disease or non-acute injury. And if you would make a subtraction from the total troponin T minus the one that is typical for chronic disease, then basically that I would expect, but we have no data about that yet. But it will be very interesting to investigate this to see. I think that most of the prognostic information is actually carried in the troponin that is detected by the smallest fragments.
Allan, you've been around for a long time, as I have, in this field of prognosis. What's your view?
Well, it's very interesting that there are, and important to remember, that there are a panoply of different mechanisms by which troponin can be released: acute stretch, supply-demand imbalance, direct involvement, exercise, etc. And we don't know which of those is predominantly involved in which of the troponin signals that we see often. For example, we know that troponin T is much more apt to be elevated in renal failure patients. All right, gives you a better long-term total mortality signal, but a less good short-term or less long-term cardiac signal, as a typical example. Now, is that due to difference in mechanisms in terms of which forms are being detected? Does this represent, in part, lack of specificity, troponin T for the myocardium, with I having better specificity? One could argue for that based on some of the skeletal muscle data. So I think there are too many different variables here to answer the question directly. And I think we need to be circumspect in not making this just a narrow, this is just ischemic heart disease versus renal failure or something of the like, which is a good use case to start with, but can't be where we finish.
Thank you, Allan. Right, I'm going to move on to some questions from our audience. I've got one from Emmanuel Otto, I think that's his name. Is troponin elevated after strenuous exercise? Well, the simple answer to that is yes. And could we have some comments from Stephen and Sarah on that one? You're on, Sarah.
So yes, definitely it is increased after strenuous exercise. But I think Stephen's group was the first to show that after strenuous exercise, it is the small forms that are producing the troponin signal there.
Stephen?
I can confirm what Sarah said. It is indeed elevated, and what Alma Mingels in our group showed is that as far as we could demonstrate it with indeed admittedly less sensitive techniques, is it appeared to be the smallest fragments. Yeah, that's it. Yeah.
Well, I think it's worth noting though, and I noted it just quickly, that the general sense, if one looks clinically, the signal seems to come from the RV with exercise release. And the thought is that one has back pressure because of increased LV filling pressures, back pressure into the pulmonary artery, and then acute impedance loading of the RV that causes this release, whether it is of I and T, and it likely represents, and may be the use case that I was suggesting we need to be careful about, what's happened when you have apoptotic release and cells die due to apoptosis. There may be very different fragments that are illuminated there than in the ischemic heart disease circumstance.
If I may add also, I wouldn't be surprised if there would be two mechanisms possible. One would be a physiologic response, for example, in healthy athletes, with a mechanism that may resemble what Allan describes. And maybe also there's a second group where the heart is actually challenged, and where exercise, like in a stress test, can be a trigger to actually induce ischemia and maybe trigger the release of a different size of troponin. So I think the new assay that Sarah discussed could also be an opportunity to better understand the whole process of exercise-induced troponin release and whether there is a pathological and a physiological variant. That's just hypothetical thinking, but that is not. That is something that is very well conceivable, in my opinion.
Okay, thank you. There are a couple of questions that have been posed on AI, and I think I can answer that very simply. At this stage in the accumulation of data, I don't think there's any. We're stuck with human intelligence because I don't think the artificial intelligence is really going to have a big enough data set to play with at this point. Another question from Martine. So in the recent era, the commercial troponin T assay measures what properties? If I understand correctly, basically this is measuring the stable fragments, isn't that so, Stephen?
Could you please repeat it once more? I couldn't understand very well, Paul. Sorry.
Okay. the question was, in the recent era, the commercial troponin T assay measures what properties? And I think we're talking about, basically, this is measuring the mid molecule here and essentially picking up just about every bit of troponin that's around.
That is correct. The clinical troponin T assay from Roche targets the middle part of the molecule. The catcher detector antibody are very close to one another, and even when it fragments, even when the N terminal is cleaved off, even when the C terminal is cleaved off, it detects actually all the troponin that well, as much as is thinkable. Yes, that is true. It is. Yeah, I think that is true.
Next question I have is saying, well, if it's true for troponin T, is it also true for troponin I? And I think Sarah covered that. And the answer is fundamentally yes, though perhaps not in the same extent. Sarah.
Yeah, so troponin I can be degraded, but can it be used similarly as we have used our troponin T assay? That remains to be seen. And I really, as I mentioned in my talk, it's what is making it complicated is to consider what we use as a reference there. What would be, as for troponin T, is easy to use the Roche assay as the reference because that's the one there is. But for troponin I, there are already assays detecting so many, or they have antibodies detecting so many different parts of the troponin I. So it's hard to show that, okay, there are the current assays and then there would be something completely different, as the current assays are already measuring different forms likely.
Well, I think you raised an interesting question of cross comparison of the different assays which are looking at different forms in terms of how they might compare in terms of outcome prediction. This could be quite an interesting thought.
But overall, and from the practical point of view, as you indicated first, if one could develop an assay for free troponin I chains, the basic substrate is similar with the clinical caveats that were indicated that I indicated when I started out.
Okay, I've got an interesting question here from Jarrett Longford, Lumira, saying the focus today has been on TNT. However, a similar approach has been taken for the IC complex, especially in response to samples with autoantibodies. Do you foresee a more universal calibration approach if more companies use the multi-antibody label and/or capture approach? I'm not quite sure I know where that's going myself, since there's been much debate about calibration of assays for quite a while. Any of you like to come in on that one?
I think I'll simply say that I agree with Fred Apple that it is unlikely that we will have a reference material that allows this to occur in his lifetime.
Thank you, Alan. I think I side with Fred and Alan on that one. Okay, it's one for you, Sarah. If you can't use serum, what do you use?
Well, for my assay, the preferred material is heparin plasma. There it seems to work fine. Also, other plasmas can be explored if they work well. For plasmas, there can be, well, also, well, first of all, it has to be confirmed that the troponin T is not degraded in the material, but also there could be limitations related to the actual assay technology used. For example, the label technology, which materials it allows. But for us, heparin plasma works the best.
What about EDTA plasma?
Well, yes, we have done some investigation with EDTA plasma. It seems that, well, it's giving a little bit lower results, but it seems that probably it could be okay, but more research is required on that.
Please remember, Paul, that and everybody, that acute myocardial infarction is a clinical circumstance with very high thrombin activity, much of the time. Therefore, the thrombin issue in regard to troponin T is not simply covered in vivo by the way in which we collect samples. There could be an in vitro component to whatever it is that is going on and modifying these forms that needs to be taken into account.
I think you mean, Alan, that even if you collected a perfect plasma, there might be activity in the bloodstream itself. Is that correct?
Well, there is. It's been well documented. There is very high thrombin activity in the setting, in particularly of ST elevation in MI, and certainly in non-ST elevation MI. If you have high levels of circulating thrombin and it is interacting with the troponin fragments that are in the circulation, then no matter how pristine your sample is, once you get it out, it has been confounded by the internal milieu, and that's important.
Yeah, there may be in fact an in vivo effect, is what you're saying. Okay, but I've got someone from darkest Peru that said that Ants wants to ask one for you, Stephen. In your experience, when we have methods that do not diagnose myocardial injury and only detect ischemic myocardial damage, and is the answer never?
Well, the topic that we covered today is if, well, there's a lot of experiments that still need to be done in that regard, but it is that the way I foresee that could discriminate in some way between acute and chronic injury. Really targeting ischemic damage is a different topic, because that would only require the detection of troponin in the presence of hypoxia, and all other cardiac injuries we would like to remain undetected. We have some work about that going on, but it is too preliminary, I think, to comment on that right now. It is very challenging, but it's too preliminary to comment on that.
Okay, next question from Peru is for Sarah. In her experience, what percentage of the methods on the market for ultrasensitive troponin use intact troponin, primary and secondary fragments in their development? So I think they're sort of saying, what are they detecting?
The current methods, what they are detecting, the troponin T.
I think this is for really for troponin T and troponin I suspect.
Well, yeah. So as has been discussed, the current troponin T assay is detecting kind of all the major forms that there are, the long forms and the short forms. And the troponin I, that is complicated depending on the antibodies that have been chosen for the specific assay.
Do you think that's one of the things that contributes to the problems of standardization?
I definitely think so.
Yeah. Next question, does thrombin also degrade troponin I? I think that's for you, Stephen.
Again, I'm really sorry, but I didn't hear the question. There's something with a bad connection.
Does thrombin degrade troponin I?
I don't know. I do know that it is the trigger to cleave the N-terminal of troponin T, and it is the presence of thrombin is something that can affect your sensitivity of the assay. That I'm really certain about. My personal experience with troponin I is definitely less than Sarah's, I think. So I don't know. I don't know.
Right, Sarah, you're on the spot.
Oh yes, but actually, I have to say that I'm not aware which are the enzymes that are cleaving troponin I, as my work has been focusing on troponin T.
So there we are, everybody out there who's listening. Here is your opportunity to strike a blow for a new area of assay development on troponin I and investigate whether thrombin is useful. I've got a question here that says, how does the high sensitivity troponin T assay relate to the 99th percentile? I think we covered that actually. It's basically the 10% CV should be below the 99th percentile. Okay, and from Guillaume Lefebvre, is the ratio of long to total troponin expressed in picograms to per picograms, or as a molar ratio? I think that's you, Sarah.
Yes, the ratio, well, depending on the units which the assays use, we use nanograms per liter for our long troponin assay, and also the Roche assay, we use nanograms per liter. And then when we do the ratio, it doesn't have any unit anymore.
But they are equimass, so to speak.
Yes, okay, that is actually an important point, because there can be some differences in the calibration of these two assays. So maybe some listeners noted that I actually showed in the figure where I showed the degradation in time. There you could see numbers above 1, like 1 meaning 100%, and then there were like 1.5 or 2. So actually, they are probably not equimolar, because that 2 would mean that there is 200% of long forms. So there is this discrepancy, but I think still it's easier to comprehend what the ratio is if you consider that as a fraction of the long forms within total.
Okay, thank you. There's a slightly divergent question here. In view of macrotroponin interferences on CT&I, will high sensitivity TNT be the preferred assay in future? Well, I regret to inform the questioner that there is macrotroponin for both T and I, and also everybody's nodding their heads, and I would actually draw their attention to an excellent article prepared by the IFCC Clinical Biochemistry group on the whole question of macrotroponins, and thoroughly commend it to them, and it's free. So, next question. The troponin T data presented compared specificity in end-STEMI patients and end-stage renal disease. High-sensitivity eye assays do not have the same performance issues with end-stage renal disease. Have you compared the same cohort with a high-sensitivity eye assay? And I think the answer to that is probably no. But I would also draw the questioner's attention to the fact that you do get elevation of troponin I in renal failure, but just not quite as much. Allan, have you got a comment on that?
Yeah, there's a nice paper by the group from Hennepin in clinical chemistry looking at the comparative prognostic influence of troponin I versus T. Both are elevated, but they're far more frequently increases in troponin T than troponin I, perhaps because the small fragments continue to be detected, and whereas with troponin I, perhaps those epitopes have been in some way degraded and are no longer present. But the prognostic significance actually for cardiovascular events is actually a little bit better than for troponin T, which gives you a better signal for overall mortality. Again, a discrepancy that is in search of a parsimonious explanation.
There have been many explanations, Allan, but I certainly agree they're not parsimonious in words. I've got a question here saying, what's the importance of troponin T and NT-proBNP for diagnosing any cardiac disease? And I think the answer to that could be summarized in yes, they do, and they detect most of it. Allan, I think you might have some comment there.
Well, I think it's worthwhile noting that we have come to appreciate that even low values that putatively would still be within the normal range, have been found to have long-term prognostic significance. And as a matter of fact, if one looks at the new guidelines from diabetes associations, they recommend the monitoring of both high sensitivity troponin and of natriuretic peptides with intervention when those values are above some of the cut points that have been reported in the literature. And those cut points vary between American and European guidelines. But we now do know that whether or not those patients have occult disease that wasn't detected, or simply this is a marker of reduced reserve in some way, or what would be called stage A heart failure, that they do presage long-term adverse events. So yes, and this is a field that is growing rapidly and will become terribly important for clinicians soon.
Okay, next question is, it's a little unclear. What is the difference between plasma troponin levels of patients with MI and CKD? I think the short answer is that they're much higher in MI than they are in CKD, but they are quantitatively different. And I think I'm going to ask Stephen to his comments on that one.
So if I understand well, looking at the absolute concentration in MI versus CKD, is that the question? Is that correct?
I don't. The question is not entirely focused. I suspect what they would like to know is what is the difference in the troponin type between MI and CKD.
Well, in chronic kidney disease, the troponin T that you can detect comprises the secondary fragments. So that is the troponin T where the N-terminal and the C-terminal appears to be cleaved off. At least that is the dominant fraction. In MI, depending on the time that you sample, but especially when you would sample early after presentation, you see also the longer fragments, so with only the N-terminal cleaved off and intact troponin, and sometimes also in combination with TIC complex. So that would be the main difference.
Okay, thank you very much. Specific question here: has the troponin composition been tested in Takotsubo syndrome? I don't think so. Anybody know?
No, but the problem
yes,
is,
unpublished, yes.
So the answer is yes, but wait and see.
Well, the problem is one of sensitivity, because in Takotsubo's troponins are not terribly elevated by and large, and therefore you're looking for a small percentage of what is a relatively lower signal. So it's much easier to start to measure this in groups that have higher signals. So that's been a problem, and several of us have over the years tried to overcome unsuccessfully.
Okay. I've got a question about Aptamer technology, which we'll answer offline. Which form is the measurable troponin in healthy individuals that we see with the current high sensitivity assays? That's for Stephen and Sarah, I think.
It's the fragmented form, secondary fragments.
Sarah, are you agreeing?
Yes, I would assume so. So the difficulty there is the sensitivity of the assay, but yes, I would agree with Stephen.
Okay, next question. Performance of cardiac troponin assays as a prognostic marker in chronic disease. That's you, Allan, I think.
Give me that again, Paul. I'm sorry, I missed it.
The performance of cardiac troponins as a prognostic marker in chronic cardiac disease.
Well, by and large, most of the data would suggest that troponin I gives one a better signal for cardiovascular events, troponin T a better signal for overall mortality. Both, therefore, may be valuable, but perhaps for different reasons.
Right, thank you. Okay, well, I've only got a couple of minutes left now. So I'd just like to reassure people who've sent in questions which haven't been answered. All the unanswered questions we will respond to by email, but we're just about at the end of our time, I think. Now, is that not right, Smeralda?
Yes.
Okay, so I would like, therefore, to take this opportunity to thank Allan and Sarah and Stephen for a really absolutely masterly presentation. I think it's absolutely fantastic, really good. And it's also, remember to be available offline, and it will be available on the IFCC website. So tell your friends all about it if they want to learn about troponin fragments. And just to reassure you, we will be responding to any questions which we didn't answer first time round, and finally, of course, thank you all for attending. So a very a good afternoon, evening, or morning to all of you.